Third, chemical modification. Chemical modification is another important post-translational modification involved in eukaryotes. This type of modification involves incorporation of small chemical groups such as acetyl, methyl or phosphate to the amino acid in a protein. Chemical modifications enable the protein molecules to perform specific biochemical function. Most important types of chemical modifications are glycosylation and addition of lipids. Let's discuss the details of these concepts. A. Glycosylation. It is a complex type of modification in which large carbohydrate side chains are attached to polypeptides. There are two general types of glycosylation. O-linked glycosylation. It is the attachment of a sugar side chain via the hydroxyl group of a serine or threonine amino acid. Please see the figure on screen. N-linked glycosylation. It involves attachment through the amino group on the side chain of asparaginase. B. Attachment of lipids. In eukaryotes, the protein molecules targeted to the plasma membrane are modified by the attachment of lipids to the growing polypeptide chain. Generally, there are three types of lipid additions: A. N-myristoylation, B. Prenylation, C. Palmitoylation. Now, N-myristoylation. The process of attachment of fatty acids to the N-terminus of the growing polypeptide during translation is called N-myristoylation. Now, Prenylation. It is a process of attachment of lipids on nascent protein molecules. In prenylation, a specific class of lipids called phenyl groups are attached to the sulfur atoms in the cytosine residues of a polypeptide chain. Many plasma membrane associated proteins involved in the control of cell growth and differentiation are modified in this way. Now, palmitoylation. In the third type of fatty acid modification, palmitoylation, palmitic acid, a 16 carbon fatty acid, is added to sulfur atoms of the side chains of internal cysteine residues. Now, Intein splicing. This is another type of post-translational processing. This process is almost similar to intron splicing seen in pre-mRNA. Inteins are internal segments of proteins. During Intein splicing, the internal segment of proteins are removed soon after translation, then the two external segments or exteins becoming linked together.